This is my investment thesis on SpliceBio, a stealth mode gene therapy startup that just raised $135 million and whose tech could unlock hundreds of billions of dollars in value across dozens of debilitating diseases. Company and platform overview: SpliceBio, headquartered in Barcelona, is engineering next-generation gene therapies using intein-based protein splicing. The value proposition: a platform approach to large gene therapy, splicing together large genes with small AAV vectors. SpliceBio uses a dual-AAV system to deliver to halves of a gene and then reconstitutes the full therapeutic protein via engineered inteins that splice the fragments in vivo. This creates a scalable, modular platform applicable to a range of diseases caused by large gene mutations, positioning the company for platform-level FDA designation if early trials show consistent efficacy and safety. The FDA platform approval possibility: the Sarepta parallel. Sarepta Therapeutics received FDA accelerated approval for multiple Duchenne muscular dystrophy DMD exon-skipping drugs based on 1. Shared mechanism of action, 2. Consistent delivery technology, and 3. Biomarker response. If 𝗦𝗣𝗟𝗏 proves safe and effective in Stargardt disease, SpliceBio could pursue a similar strategy. 1. Seek FDA engagement for a platform-based regulatory path. 2. Use Stargardt data as a foundation to accelerate other programs, for example, Usher syndrome, Batten disease, and 3. Reduce time and cost to market for pipeline programs by leveraging platform validation. Market pull plus technology push to key competitive advantages. Market demand: Stargardt disease affects an estimated 1 in 8,000 to 10,000 individuals globally, with no approved treatments. Young onset, progressive blindness, and lifelong visual disability create a high-cost burden and strong reimbursement potential. Patients are often diagnosed in adolescence, creating opportunity for long-term QALY improvement. Technology differentiation: SpliceBio solves AAV size limits without requiring new delivery systems, retaining regulatory familiarity with AAV, which should improve FDA engagement prospects. It also offers a plug-and-play architecture to rapidly apply inteins to other large gene diseases. Real-world evidence and economic impact: if successful, 𝗦𝗣𝗟𝗏 could preserve or restore vision, adding an estimated 5 to 10 QALEs per patient, with the lifetime societal costs of blindness ranging from $1 million to $3 million. Payers may support premium pricing backed by outcomes-based agreements. Compared to supportive treatments or assistive technologies, SpliceBio's one-time therapy could offer superior cost-benefit outcomes over 10 to 15 years of life. Competitive landscape: SpliceBio leads the niche of large gene retinal gene therapy using a clinically validated AAV platform. Key differentiators over peers: 1. CRISPR-based competitors, for example, editis, durable edits, but delivery and safety concerns remain. 2. Non-viral systems, for example, mini-circle DNA, high capacity, but immature clinical data and complex translation paths. 3. HSV1-based systems, for example, e.g. for 𝗦𝗘𝗔𝗣, high payload, but risks include immune reactivity and scalability issues. SpliceBio's advantage lies in its balance of innovation and regulatory familiarity, making it a first mover in a platform race for large gene therapies. Financing, clinical strategy, and platform scaling: Series B, $135 million. Raised in June 2025, co-led by EQT Life Sciences and Sanofi Ventures, joined by Novartis, Roche, NEA, UCB, Eisaios, Gilead, and Asebis. Capital enables: 1. Completion of Astra Phase 1 to trial Stargardt. 2. Advancement of second candidate pre-IND expected 2026. And 3. Expansion into 1 to 2 additional indications using same intein AAV framework. Clinical plan and regulatory path: 1. Astra trial 𝗦𝗣𝗟𝗏, FDA and MHRA approved. Dosing begins February 2025. Polaris observational study, capturing real-world evidence visual function metrics, and call baselines. 3. FDA interactions. Leverage Stargardt program as template for other large gene disorders under a shared technology master file. Risk-adjusted DCF valuation 20-5-2030. Conservative case single indication. 50% pause for SB007. $200 million peak global revenue. 10% royalties or outcomes-based contracts. 15% discount rate. Estimated valuation, $150 million, $300 million. Platform case multi-indication plus FDA platform path. Stargardt plus 2 to 3 follow-on indications, $1 billion to $1 billion combined peak sales. Platform efficiency cuts time to market by 40%. Valuation by 2028 to 2030. $1.2 billion to $0.5 billion, with 5 RWA for Series B investors. Exit strategy and EPS outlook, IPO window. If SB007 data are positive in 2026 to 2027, SpliceBio may pursue a 2027 IPO on the strength of 1. A validated platform, 2. An expanded pipeline, and 3. A strong strategic syndicate including pharma VCs, acquisition or licensing. Roche, Novartis and Sanofi may explore M&A or licensing deals, particularly if a platform-level approval strategy is validated. CVRs contingent value rights. Any deal may include CVRs tied to milestones such as Phase 3 initiation, FDA platform designation, and approval of a second indication. Commercialization roadmap and catalysts. 2025 First dosing in Astra trial Stargardt. 2026 Initial efficacy readout. Pre-IND for next indication 2027 Pivotal trial planning. Potential platform discussions with FDA 2028 Platform-based IND filing for next 1 to 2 diseases. 2029-2030 First product approval plus possible IPO or M&A exit. Final outlook. SpliceBio is building a high-leverage, high-potential gene therapy platform, solving one of the industry's critical bottlenecks: gene size limitations through a clinically validated, modular approach. If SB007 succeeds, SpliceBio could follow Sarepta's path and unlock multi-asset acceleration via FDA-endorsed platform designation. This would substantially compress time and capital needs for future therapies and could deliver significant upside, making SpliceBio one of the most promising stealth mode platform companies in genetic medicine today. Thanks for watching. See you next time.